Hello, my name is Tom Zimmerman. I'm the director of the Division of Laboratory Animal Resources. We provide the husbandry and veterinary care for all of the animals used in research here at Stony Brook campus. I'd like to talk to you today a little bit about the use of animals in research. We'll talk a little bit about the history of animals used in research, the early laws that governed their use. Then we'll move forward to the present day. How do we use animals now? What are the current regulations? Who enforces those regulations? What happens when problems are found? And what do you as a researcher what role do you play when you're using animals in research? So the first recorded use of animals in research was back in the 200 BC. First was Aristotle. This is the written record of animals used in research. Aristotle was in the record. He has made many contributions to science, but especially in the field of biology. What he did with his animal dissections was essentially divide animals into those with blood and those without blood. And later we came to know those as vertebrates and invertebrates. A little later, Erasistratus was known for actually identifying that the heart was not the center of sensation as was previously thought, but was actually a pump, and he accurately described the heart valves. And he actually had theorized that this pump, unfortunately, was pumping air, which carried the animal spirit pneuma, and then this pneuma was transferred to the cells through the axons and the neurons. So he had a little bit to learn yet. Then, of course, everybody is familiar with Louis Pasteur and his work with the germ theory. He used anthrax bacteria in sheep and was actually able to transmit the disease from one sheep to another, proving that, as was previously thought was erroneously wrong, that diseases are not spontaneous, but they can be transmitted from animal to animal. Not long after that, I'm sure you're familiar with Ivan Pavlov and his use of dogs to demonstrate classical conditioning. He was using the bell and linked it with saliva production at mealtime. And you can see one of the dogs there has a test tube actually sutured to his cheek to collect the saliva during the experiment. And who can forget the first chimpanzee in space? That would be Ham in 1961. He was the first hominid to actually go up in space, leave the Earth's atmosphere. His capsule was up for a total of 16 minutes, and he successfully landed, much to everybody's amazement. One of the things that's interesting is he was known as animal number 65 until he successfully landed. The people at NASA didn't want to have a named chimpanzee that might be reported in the news if the mission was not successful. And then after that, a rather strong breakthrough in science came with Rudolf Janisch in 1970s, where he was able to do the first genetic manipulation and inserted some genes from the SV40 virus, which normally affects humans and non-human primates. He was able to successfully insert them into the genome of the mouse. And so this was the first transgenic animal that was created. And his work basically opened the doors for a lot of other genetic engineering techniques. Not only now can we insert foreign DNA into an animal, but we're also able to selectively knock out genes that we want, or we can conditionally knock them out so that we can have them expressed to a certain point in the animal's life, and then we give something like tetracycline in the water or Taxol injections, and that will then excise that gene, and you can look at then what that gene actually does in the animal's anatomy and function. And then the genetic engineering finally led to one of the great breakthroughs was Dolly the sheep, who was the first animal who was successfully cloned from a somatic cell. Dolly was generated by taking basically a cell from the mammary gland of a sheep, taking an egg from a sheep, taking out that nucleus and doing nuclear transfer from the somatic cell into the egg. That was then implanted into a surrogate mother, and Dolly was born. Interestingly, this happened in Scotland, and the people had a bit of a sense of humor. Dolly was named, since the origin was from breast tissue, they named her after the curvaceous singer Dolly Parton, and so that's where she got her name. So before I go on, I just wanted to introduce one term that's often used a lot with the discussion of animals in research, and that's the term bioethics. Bioethics is defined as the philosophical study of the ethical controversies brought about by advances in biology and medicine. Some other examples of bioethical terms might be organ donation and organ transplantation, things like abortion, euthanasia, stem cell transplant. All of these things are potentially controversial, but they are advances in biology. And obviously, using animals in research would be another example of that. And based on the individual person and the society that they're brought up with, they will have specific feelings on whether they approve or disapprove of that. And a lot of that has to do with the culture that you are being raised in. For instance, in the US, we seem to have a lot of issues with abortion or stem cell research, and much of that can be traced back to the different religions that are practiced here in the U.S. and the ties that are associated with that. So as we think about using animals in research, we have to ask the question, do we as humans have any moral responsibility for these animals? Do we have any obligations to their health and well-being? And if you think about, if you're driving down the street, you don't necessarily run over dogs and cats, and I'm sure your neighbors wouldn't be too happy about that. But so that in itself says that we, at least as United States citizens, do feel that there is some moral obligation that we have towards using animals or towards animals. And so that brings into the question, if the animals have some moral standing, are they then eligible for rights? Should they be granted rights like we do for humans? And in fact, in 2002, there was legislation introduced in the United Kingdom which would have given chimpanzees rights, the right to life, the right of freedom, the right to be free of torture. Unfortunately, or fortunately, I guess, that legislation died in committee because there was so much discussion about what would happen if. And so they're saying, well, if the chimpanzees have rights, what's to stop the animal rights activists from representing them in court and taking all the entertainment industry and the pharmaceutical industry and suing them because of how they had used the chimpanzees in the past? So that issue has died, but certainly may come up again in the future. So even back in the very beginning, this discussion was occurring about animal research and animal rights and how do we treat them. And there's a wide spectrum of thoughts on this. Descartes basically said that animals are automata and that they essentially they don't feel, they don't have a conscience, we don't have to worry about them. Somewhere in the middle is Kant and Aristotle, who believe that while they certainly can feel pain, it really is of no moral concern to us. And then on the other side of the spectrum are people like Hume, who say that not only can animals reason, but they can feel a full range of emotions and they should be treated accordingly. So as we go through history and the act of vivisection became more and more popular in the 1600s and 1700s, the populations there became divided with different feelings on either for or against the act of vivisection. There were people in the camp of Edmund O'Meara who said that no, it shouldn't be allowed for a number of reasons. One, the pain of the procedure would alter the results, so the data you're getting could not be accurate. They say that animals in general are inferior to humans, and so the results could not be translatable at all. And then there are those that bring ethics into it, saying that the benefit to humans just simply cannot justify the harm that you're doing to the animals. On the other hand is somebody like Claude Bernard. He is known as the Prince of Vivisection in his time, and as a strong advocate saying experiments on animals are entirely conclusive for the toxicology and hygiene of man, and these substances are the same in man as they are in animals, to maybe a slightly lesser degree. Interestingly, Dr. Bernard's wife was the first person to start the Anti-Vivisection Society in France. So I can just imagine their dinner table conversation with the Prince of Vivisection talking with the founder of the anti-vivisection movement. So as more and more discussion was generated around the use of animals and around vivisection, that prompted people to then think about some regulations to protect the animals. And so the first Animal Protection Act was created in 1822, and it was passed primarily to protect cattle and livestock that were being driven to market. Actually, the first law was passed in 1655. It was in Ireland. That legislation prohibited you from pulling wool off of sheep and from tying your plow to horses' tails. So we've certainly come a long way since then. So the animal protection laws in Britain morphed a little bit, and by 1876 they had passed the Cruelty to Animals Act, which was the first one to actually regulate the use of animals in testing and research. It took about 50 years for the animal rights kind of feeling to come across the Atlantic to the U.S. It was in 1866 that Henry Bergh founded the American Society for the Prevention of Cruelty to Animals, known as the ASPCA, and it still is around today. They were the ones instrumental in creating the first animal hospitals, and they did that primarily so they could treat horses. Horses were the main form of transportation, and whenever they had an injured leg, they would have to be shot. So they created some animal hospitals and were able to start doing surgery on those patients and save those horses for future use. But it took about another, and they were also instrumental in creating state anti-cruelty legislation, mostly against dog fighting, against cockfighting, and against something called cock throwing, which I have no idea what is. But it took about another 100 years before there was any federal legislation that was passed. And so the first federal legislation was the Pet Protection Act, which was passed in 1966 and later was renamed the Animal Welfare Act. So that's a little bit about the history. So how do we use animals in research today? Well, one of the main uses is still basic biology. In fact, about over half of the research that we do in animals today is still looking at basic human and animal functions, the physiology. How do we as a being work? How do our organs interact and the processes interact at a cellular and subcellular level? There's still a lot that we don't know. And especially when you think that, for instance, Watson and Crick, it was the 1950s when they elucidated that the DNA was a helical structure and how it replicated and divided and that sort of thing. And now only 50 years later, we've already sequenced the human genome, and we're using that information now to create transgenic mice. And so for every single gene in the human, we can actually create an animal model that we can now look at the function of that gene by either knocking it out or overexpressing it or what have you by the genetic manipulations that we're capable of doing now. Another major use of animals is to model diseases. For every single human disease, there's a corresponding animal model out there. It could be either a naturally occurring disease in animals, or it could be genetically induced, again, by manipulating the DNA. It could be surgically induced or chemically induced. But you need to have some sort of model that you can work with so you can study the origin of the disease, the pathogenesis, how it progresses, possible ways to prevent or treat the disease. And most of those would simply not be possible in humans, again, because of the ethics and how we feel about what we can do to people versus what we can do to animals. Another common use of animals is in surgical models, whether it's something as simple as pinning or plating a broken arm to something as complex as doing a heart and lung transplantation. All of those procedures were first worked out in animals before they were tried in humans. And certainly I don't think I would want to be going into an operating room and having the surgeon thumbing through a textbook saying, this ought to be fun, I've never tried this before. So everything is first tested out in animals before it's used in humans. The FDA still requires any pharmaceuticals or biologicals to undergo safety and efficacy testing. In general, it has to be tested in at least a rodent species and a non-rodent species, and the animals have to be dosed for as long as and at levels of whatever would be given to humans before any thought can be given of actually giving that drug to the human population. The EPA also requires that any potentially toxic compounds be tested. Again, in animal models, you need to know what the organ of toxicity is. You need to know what levels are poisonous to humans. You need to know potential anecdotes, personal protective gear that you should wear when handling it. All of these things are, again, first worked out in animals before they're translated to the human population. They're also used to some degree still in training and teaching. Med students, veterinary students, people learning microvascular surgery, people in undergrad labs like pharmacology or biology or anatomy, all still use either live or dead animals as part of their curriculum. And a couple of advantages of using animals over humans. One of them is the ability to tightly control all of the environmental parameters. All of our laboratory animals are housed the same way, have the same food, the same bedding, the same light cycle, the same room temperature, humidity. All of that is exactly the same. Whereas if you were working with a group of 50 humans, there is no way that you're going to be able to factor out all of those potential environmental confounding variables. In addition to the environmental factors, you can also tightly control the genetic variables. There are many rodent strains that are inbred, which means that they are 99.9% genetically identical to their mom and their sister and their brother. So you can get large populations of genetically identical animals, keep them all in the same environment, and essentially you've taken out all the potential confounding variables that would be involved in your experiment, especially if you were doing something in humans. In addition, you're able to obtain large numbers of populations of animals. The typical rodent gestation period is 21 days. They have 8 to 10 per litter, so you can see in a short amount of time you can get 50 or 100 animals, all the same age and all the same genotype, and able to do a large study very easily. So how many animals do we use? It's estimated by the National Academy of Sciences that every year we use between 50 and 100 million animals in research. And while that may seem like a lot, to put it in perspective, that really is less than 5% of the animals that we kill each year for food. And in addition, over 95% of the animals are mice and rats, and less than 1% are cats, dogs, and non-human primates, which seem to be the most sensitive species when you're talking to the animal rights groups. The total number of animals used in research has been consistently dropping over the last 25 years, although that's now starting to change a little bit. Again, with the genotyping and the genetic manipulations, we're now creating mouse strains for every single gene, and so you can just imagine how many different mouse strains that's going to take to actually drill down and dissect what each gene does. And about 99% of the animals that we use in research were purpose-bred. They were born to go into research. There are vendors that sell rats, there are vendors that sell dogs and cats, and they are bred specifically for use in the laboratory. The one exception to that are there are a few species of non-human primates which are still wild-caught, but even that number is decreasing every year. So that's the use of animals. What are the regulations that we have to follow if we are going to use animals in research? Well, as I mentioned, the first legislation was the Animal Welfare Act, which again was called the Pet Protection Act, was passed in 1966. This is enforced by the United States Department of Agriculture. And the way this came about was there was a dog that lived in Virginia, a coon dog whose name was Pepper. And Pepper lived on a farm in Virginia, and Pepper disappeared, and the owners were thinking, that's strange because Pepper usually stays close to the home. Didn't think much of it, but about 3 or 4 weeks later, there was an article that appeared first in Sports Illustrated, and then a couple of weeks later, the same topic in Life magazine. And these were exposés about people that were living around the country and going around and buying up dogs and cats, either from people, going to flea markets, answering ads in the newspaper. They would buy up these dogs and cats and then turn around and sell them to research facilities. And indeed, that is what happened to Pepper. Pepper had been stolen from the farm, turned around, gave him to one of these pet rustlers who had then turned around and sold it to a research facility in upstate New York. And indeed, Pepper had been used on a cardiac pacemaker study, was anesthetized, they were implanting a pacemaker, and she died on the table. And so all of this was in the article. So the family wrote their congressman and said, why isn't anybody watching out over these people? Prior to 1966, there was no regulatory people overlooking circuses, zoos, pet stores, puppy mills. All of that was free game. So the care that the animals got was basically dependent on the person giving the care. And some people were good, some people were not so good. So they passed the Pet Protection Act, and it's been amended a few times since then. And since it was the Pet Protection Act, it does not cover mice, rats, or birds, which, as I just told you, is about 95% of the animals that we use in research. However, there's a second set of laws known as the Public Health Service Act, which is enforced by the Office of Laboratory Animal Welfare. Anyone who's receiving PHS money, that's government grant money, so that would be money from NIH, NSF, FDA, CDC, anybody receiving those funds has to abide by the Public Health Service Act. And so Stony Brook gets about 2/3 of its money from PHS funding, so we do have to abide by these guidelines. And they do apply to all vertebrate animals, including mice and rats and birds. So basically the act is summarized in something called the Guide for the Care and Use of Laboratory Animals. And that is our Bible. That is what we follow in our day-to-day care of the animals. And then there's a third regulatory body known as the Association for the Assessment and Accreditation of Laboratory Animal Care International, or ALAC International. This is a group of about 650 laboratory animal veterinarians that volunteer their time. And every 3 years, we as an accredited facility have to send them a description of our program, our animal care and use program, which basically describes how do we at Stony Brook, what policies and procedures do we have in place to make sure we are obeying the Animal Welfare Act and the Public Health Service Act. And so they make sure that we are obeying the laws, and they also will come and site visit every 3 years, and they look at our program, they inspect our facilities, they visit our Animal Care and Use Committee, and they make sure that not only are we obeying the law, but are we also keeping up with other institutions in the U.S., keeping up with the lab animal as an industry in general. panel? Do we use isoflurane anesthetic now instead of ether? Do all of our post-operative patients receive some sort of analgesic? 5 or 10 years ago, we used to do surgery on rodents and not think anything about aseptic technique or providing pain relief afterwards. And as a result, many of those studies are not able to be reproduced because pain does have an effect on your results. So those are the main regulatory bodies that we have to follow. And essentially, in the Guide for the Care and Use of Laboratory Animals, which is from the Public Health Service Act, it basically spells out everything that we as an animal resource have to fulfill, from the size of the cage, how many square inches does a mouse need, or how many square feet of run space does a dog need, how often do you have to feed them, how long is the feed good for? Every 6 months we have to rotate the feed. The temperature acceptable ranges in the room, the humidity ranges, how hot does our cage wash temperature have to be to adequately disinfect our cages, how often do we have to disinfect the cages? All of that is spelled out and must be followed, and those are the things that the inspectors will look at when they come to take a look at our facility. One of the amendments to the Animal Welfare Act now states that use of dogs in the laboratory, they have to be provided with an opportunity of a regular exercise program. Non-human primates have to be given an environmental enrichment program, which helps to stimulate their species-specific behaviors. It also says that any procedure that might cause pain or distress has to be accompanied with anesthesia or analgesia, and that any surgery must be done using sterile technique. And it also says that anyone working with animals has to receive the proper training before they can lay a hand on them. And that's part of what we do here in DLR and at Stony Brook, is provide training to all the people who are using animals who have never had to use animals before. And finally, the regulations require that each facility have what's called an Institutional Animal Care and Use Committee, referred to as the IACUC. And as you'll see, it's a very powerful committee, probably one of the most powerful committees here on the campus. And it's by law, this committee is made up of at least one scientist, that's somebody who's practicing research at the time, at least one layperson, that's somebody not associated with the university and not associated with research. We actually have a minister and a school teacher on our committee to serve as and they represent the outside interests, basically the community interests. And then you have to have at least one veterinarian who's had some sort of training in laboratory animal medicine. So it's up to this committee to basically review and subsequently approve all the protocols that are going to be using animals here on campus. Before you can do anything with an animal, you have to fill out the animal care and use form that basically makes sure whatever you're going to be doing is consistent with the regulations. In addition to reviewing the protocols on a monthly basis, every 6 months we also go out and inspect the laboratories on campus that do animal work. One of the questions in the form is, are you taking animals out of the DLR facility, the lab animal facility, and if so, where are you taking them? And we make a list of all of those places, and every 6 months we go around to the labs and make sure that that even if you're not working in DLR, you are indeed obeying the regulations. So if you've got animals in your lab, we make sure they have clean cages. Is there food? Is there water? Is there a log to show that they've been checked every day? If you're doing surgery, do you have a proper surgical records? Are your instruments sterilized to start with? If you're using controlled drugs, are they properly locked up? All those sorts of things are checked on every 6 month basis, again, to make sure that you in your area are obeying the regulations. And in addition to the laboratory inspections, there are other forms of post-approval monitoring. So once the protocol is approved, it's up to the IACUC to make sure that whatever animal work is going on is consistent with those protocols. And so one of the things in the protocol is how many animals are you using? And so we keep track of how many animals are ordered for each protocol. And as you get close to your number, we say, hey, you're going to run out of animals. We also will look at, if we come to your lab and you're doing surgery, then we will look to see your surgical notebook and look to see the patient, look to see your post-operative records. So again, post-operative monitoring is just as important, if not more important, than the initial approval of the protocol. And there is a requirement in the regulations that the laboratory animal veterinarian must be involved with the protocol. So we recommend that if you are involved with writing any of these protocols, that you actually submit a draft to the veterinarian so he has a chance to look at it, make sure everything's in line with the regulations before you go to the trouble of submitting it to the committee just to have it rejected because you didn't take into account certain things. So the protocol itself is a form that basically includes all of this as far as information that's requested. The personnel, you have to list who's going to be doing the animal work, what sort of training do they have. The study has to be justified. What questions are you trying to answer? What good will it do for mankind and humans once you've conducted this particular experiment? The animals have to be adequately considered. The species that you're using must be appropriate for the model that you're trying to recreate. The numbers have to be based on statistics. They can't just be drawn out of a hat. You need to use enough animals so that you prove your point and don't have to repeat the study, but you don't want to be using so many that you're wasting animals and using some unnecessarily. You're required to do a literature search for alternatives, either to alternatives to animal use or alternatives to painful and distressful procedures, depending on what your protocol is. And then probably the most important part is the details of the procedures. Everything you do from the time the animal comes to our facility until the time it's either euthanized or transferred to another protocol has to be explicitly described in that protocol. And that is what the committee reviews to make sure that it is in lines with the regulations. If you're doing anything invasive or doing surgery, you have to have the proper anesthetics and analgesics available. With what substance are you using, what dose, what's the frequency of administration. For each of the procedures that you've detailed, you need to think about what might go wrong, what could be a potential adverse effect, and if it occurs, how will I treat that animal? Most of these studies end up with euthanizing the animal and taking tissues and examining them at the very end. So what is the method of euthanasia? Again, it has to be one of the approved methods according to the regulations. If you're using hazardous agents, what sort of precautions need to be taken, both for the person handling the animals, what kind of personal protective gear do they need, as well as the animals themselves. What side effects might they see? Do we have to handle their bedding and cages any differently? All of that stuff has to be spelled out before you can even start doing the animal work. And one point to note is that the IACUC, the Animal Care and Use Committee, does not review scientific merit. They leave that up to the institution providing the funding, either the NIH, the NSF, whatever that may be, or if it's funded privately, then the scientific merit is reviewed by the department chair. So that's not something that's evaluated by the committee. So those are the regulations. So who enforces them? Well, as far as the Animal Welfare Act, I told you it's the United States Department of Agriculture. Every year they come unannounced, come knocking at the door, usually the first week in January, right after the new year, and they spend 2 or 3 days at our facility interviewing the IACUC, talking to researchers, going around looking at the animals, looking at the facility, looking at all of the animal records and that sort of thing. The state law is enforced by the New York State Department of Health. They also inspect once a year, although they usually make an appointment, so they make sure that we're there. The PHS Act is enforced by the Office of Laboratory Animal Welfare. They generally will not routinely inspect unless there's a problem that has been reported to them. And then locally, as you've learned, the Institutional Animal Care and Use Committee is the main thrust behind enforcement of the regulations, and especially because they're at a local level, they see what's actually going on. And so both by the program review and the follow-up, the post-approval monitoring, then they basically enforce what has been written in that protocol. So what happens if problems are found? Well, for the USDA, when they come and do their annual report, they essentially will do an exit report which lists each of the problems as well as a citation from the Animal Welfare Act of what problem, what the problem was as far as going against the regulations. So if it has it might say chip paint, and the regulation will be all primary enclosures must be kept clean and neat and sanitizable, or something like that. So you get a list of these different findings and a list of the regulatory verbiage that is associated with those. And keep in mind that these documents are then filed and are open to the public. They used to have to be requested by FOIA, requesting documents officially when they were paper copy. Now they are going immediately onto the internet. So as soon as we have our inspection, our institution results are now posted on the internet, and that is something that the Animal Rights Group monitors very frequently. And in fact, you can see here a recent article is discussing findings that were found at Covance facilities. And again, they were looking at inspection reports and zeroed in on this facility because there were problems that were published. The report, in addition to the problems that were found, will also include a date that you need to have the problem fixed by. And they will oftentimes come back and re-inspect to make sure that the remedy has been put in place. And depending on the severity of the problem, they may impose fines. If an animal has died or you've put an animal in danger of being harmed, there may be fines, or they may up their inspection schedule so they come every 3 or 4 months until they're sure that your program is back on track. And finally, the IACUC, depending on what they find during their post-approval monitoring, they may report findings to the USDA that are basically self-identified findings. So if we find somebody who's going off a protocol and it is a species that's covered by the USDA, we are obligated to report that to the USDA. And as part of reporting, they also request that you include some sort of plan to prevent that problem from occurring in the future. So what about OLA? What do? The Office of Laboratory Animal Welfare. Well, they're a little bit different. Each institution, like Stony Brook, has on file with OLA what's called a written assurance. And basically, it spells out all of our policies and procedures that we have in place to make sure that Stony Brook, as an institution, is obeying the PHS Act. And so we say that we're going to do it, and they trust us to do that. And so they rely basically more on the institution to self-monitor themselves. And so if we do find any problems, just like USDA, we will then report them to OLA. And again, you need to come up with some plan to prevent that from happening in the future. And usually OLA says, yes, good job, thank you very much. And they don't really tend to get too worked up about it. And again, if we suspend any studies, that also has to be reported to OLA. Now, I mentioned that they don't typically make unannounced inspections, but one caveat to that is I've talked with people in OLA, and they say while they don't generally go inspect institutions that have been reporting things, they will go out to institutions that have never self-reported anything, because they know that people aren't perfect, and they know that especially institutional research, academic researchers are known for not obeying the rules. And so if they've never had a report from your institution, they figure either your IACUC is not doing their job or they're covering up stuff and not reporting it. So those are the times when they're actually going to come out and visit you, as if you've never reported anything to them. And so what happens if problems are found at the institutional level? Well, just to recap, there are three major regulatory functions of the IACUC. Obviously, reviewing all the proposed animal work. So when the protocol is submitted, it's their job to go through it and make sure it's consistent with the regulations. They also then do the monitoring of the ongoing work, making sure that whatever was approved in that protocol is what's being done to the animals and nothing else is being done. And finally, we do occasionally get reports from faculty, students, staff members about potential animal abuse, and it's up to the IACUC to investigate that allegation and then come up with some remedy. So when problems are found, if they're found during the protocol submission process, this is usually the easiest thing to take care of because the protocol hasn't been approved yet. Usually when we review the protocol, we'll come back with a list of concerns or questions or things that they've missed, missing information, or things that are not consistent with their regulations. Those comments will go back to the investigator. They have a chance to then rewrite it and resubmit it, and hopefully it will pass the second time. So those are relatively straightforward. However, problems found during the post-approval monitoring period are usually more serious. They could include things like inaccurate record keeping or no record keeping. Basically, the USDA inspector says if it's not written down, it didn't happen, and we run into that all the time. People are very careful about putting the data in their laboratory notebooks, but much less careful about filling out the required forms as far as the animal records, the anesthetic records, the surgical records, and that sort of thing. So those are things that we look at during our post-approval monitoring period. Other things would be lack of proper storage of controlled drugs. As I mentioned, controlled drugs by law have to be behind at least 2 lock sets. So that could be the lock on your lab door and the lock on a desk drawer, or the lock on a desk drawer and the lock in a lockbox in the drawer, anything like that. But sometimes we come and we find some of these controlled substances sitting out on the table, and obviously that's not appropriate. Other times we find expired drugs. If you don't use a drug very often or you don't do a procedure very often, there's a chance that drug will sit on the shelf past its expiration date. And so those are the things that we look for when we go out and do our post-procedural monitoring. And obviously the worst potential problem would be not following the protocol, and that usually is the most serious if you have a protocol deviation. So when any of these things happen, what we do is the IACUC will then draft a letter and send it to the investigator asking, this is the problem that we encountered, why did it happen, and how are you going to prevent it from happening in the future? And for the most part, it's pretty straightforward, and there are no problems subsequent to that. If the problem is serious enough, then again, the protocol may be suspended and the regulatory agencies may have to be notified. One other thing that we tell people, and especially anyone going into the DLAR facility has to come to an orientation given to me. And one of the things that I impress upon them is that if anyone at any time sees what they consider animal abuse, which would be like dressing a dog up in a Santa outfit, if they see anything, either the procedure that's being done on the animals should never be done on animals, or the procedure's okay, but the person who's doing it sure doesn't seem like they're trained to do what they're doing, that has to be reported to the IACUC, and then they have to follow up on it. So they basically will investigate it. Oftentimes they'll form a subcommittee. They'll go and talk to the person who made the report. They'll talk to the people that might have been involved in the report or the mistreatment complaint, and they'll find out exactly what happened. And if indeed there was an act of animal abuse or mistreatment, then there will be punitive measures taken against the person who did it, as well as preventive measures put in place to prevent that from happening in the future. And again, depending on the problem, the protocol may be suspended and the agencies may have to be notified. So what role do you as researchers who might be using animals in research play? What are your responsibilities? Well, one thing that we tell people who are working in animal research is to always keep in mind something called the 3 R's. The 3 R's refer to replacement, reduction, and refinement. These terms were coined in 1959 by Russell and Birch in a paper known as The Principles of Humane Experimental Technique. And what they did was they looked at some animal studies for about the last 15 years and kind of looked at the ones that were the most invasive or that had the most potential to cause pain. And they thought, what can we do to help minimize the potential adverse effects that the animal will see? And they came up with these 3 words. Basically, replacement refers to only do studies in animals that can't be done using non-animal methods. If you can use a computer simulator or reagents on the benchtop, do that first. If you can't use reagents on the benchtop, but you can use bacteria or fungus to answer the questions you want, use them first. And only if you can't use the lower animal life do you then proceed on to animal methods, and you should select the species that's lowest on the phylogenetic scale. So if you can do work in lobster or crabs or shellfish versus mice, then do that. If you have to go to mammals, then you would use a mouse instead of a rabbit, or a rabbit instead of a dog. So you want to use the lowest phylogenetic animal that you can to do your research, and that's replacement. Reduction refers to the animals the number of animals that you're going to be using. And again, this should be based on statistics. You should be able to estimate, well, with this treatment, I expect a 30% difference between the treatment and control groups. And if I want to have a 95% confidence level, then I look on the chart and I should have 12 animals per group. You want to make sure that you get the right animals, the right number. So again, if you have too few, you might have to repeat the experiment. If you have too many, you're wasting them. And it's also important to keep in mind, if the procedure that you're doing, you anticipate that not every animal will make it to the end of the study. If you're doing surgery and you expect some mortality or something, you need to factor that in up front, again, so that if you have animals that drop out along the way, you suddenly don't come to the 3rd month and have to start over because you don't have enough animals to finish the study. And the last one is refinement. And this basically refers to animals that are going to be used for potentially painful and distressful procedures. And the thought of refinement is to design your study to minimize any of that impact. So if your study calls for food deprivation for 20 hours, could you accomplish the same thing with only 10 hours? Or if you're doing surgery in the abdominal area, can you do it laparoscopically with just a small pinhole incision versus opening up the whole abdomen? Or if you're doing blood sampling and you've got 10 samples scheduled over a 24-hour period, could you really get the same information by only taking 5? So those are examples of refinement, where you take what you're planning and really look at it hard and see, can I tune it down a little bit to help minimize the potential impact on the animals that we're using? In addition, it's your responsibility to make sure that whatever you're doing is in that approved IACUC protocol. And anybody who's working in your lab should be given a copy of that protocol, and they should read that cover to cover. Because again, if it's not written in there, you're not allowed to do it. You need to make sure that all the people that are working with animals have been adequately trained. Many of our undergraduate students coming in have never worked with animals before, and you can't just turn them loose on an animal study and expect them to be comfortable, much less get usable data. And finally, if anyone in your lab does bring a concern to you about laboratory animal welfare, accusations of mistreatment or abuse, it is your responsibility to then take that information and report that to the Animal Care and Use Committee so that they can follow up on that. So, how does the general public view biomedical research and animal use in biomedical research? Well, a poll that was taken just a couple of years ago determined that more than 75% of the general population does support the use of animals in research, with the following caveats. 1, the research must be necessary, and I think we would all agree to that. 2, the animals must be humanely treated. Again, no argument. And 3rd, pain and distress must be kept to a minimum. And I think you'll agree that with the regulations that I've shown you, basically all of this stuff has to be met in order to be in compliance with the regulations. So in general, I think the public is on our side, or is on the side of using animals in research, but they really just need to understand what the goals are and what the end result's going to be before they have buy-in. Now, there are, on the other hand, there are assertions from the animal activist groups that tend to counteract that thought. They say that most animal research is needlessly cruel and painful. Well, I beg to differ. It is shown that less than 2% of the animal studies in the US actually have unrelieved pain and distress. All the other ones have either anesthetics or analgesics that are administered. And the 2%, most of those studies are actually studying the phenomenon of pain and how to prevent pain or how to lessen pain. And if you're studying pain, you can't very well prevent it and still study it. So again, I disagree with that assertion from the animal rights people. They also say that research with animals is now unnecessary because there are alternative models that now exist. Again, that may be true for some things. We have been able to try computer paradigms to replace some animal testing. We've replaced cell cultures for some of the things that used to use whole bodies. In fact, the EPA now allows artificial skin to test dermal irritants. They're using now horseshoe crab red blood cells. Instead of injecting substances into a rabbit to see if they get a fever, they inject it now into these horseshoe crab cells. If they clump up, then that compound is considered a pyrogen. So we are replacing slowly some of the animal procedures with non-animal procedures. But as I told you, more than 50% of the research done is still basic biology. And there's no way you can create an animal model or a non-animal model if you don't know what you're trying to model. So there's still a lot that is unknown, and there's no way that we're going to be able to replace animals, at least in the near future, for those studies. And then their last assertion is that human health has actually been harmed by misleading results that have been obtained by animals. And again, I beg to differ. If you talk to anybody who works at a pharmaceutical company, they'll tell you that less than 1 compound out of 100 will ever make it to humans. And the main reason for that is as those other 99 compounds progress the way up the drug discovery trail, they eventually get put into animals, and if they're not bioavailable, if they're metabolized too fast, or if they cause side effects, then they get dropped out of production and they're no longer considered. So I would disagree. And in fact, some people will point to the thalidomide cases with the birth defects and saying that that was due to lack of animal testing. In reality, at the time that came about, they were not required to do reproductive toxicology on compounds. And it was not until after the thalidomide crisis and all of the deformations came out that they realized we never tested this in pregnant animals before. And when they went back and did it, lo and behold, there were birth defects. So in reality, we might even need to be testing more thoroughly before we put things into human, much less that human health has been harmed. So let me just end by a couple of different activist groups that you were probably familiar with. One is PETA, the People for Ethical Treatment of Animals. They're always in the headlines. They basically gained their claim to fame in 1980 during the Silver Springs monkey case. And how that came about was there was an investigator at Silver Springs, Maryland, who was doing some research in primates where he was severing the nerves or the sensory nerves. And so the different monkeys couldn't feel their arm or couldn't feel their leg. So there was a student named Alex Pacheco who volunteered to work in that laboratory and was working there for a few months, and then wasn't real happy with what he saw and ended up reporting to the authorities. And it wasn't about the surgeries or anything, it was about the living conditions of the animals. And so the authorities came in and investigated, and they ended up charging Dr. Taub, who was the research investigator. They had like 116 counts, and by the time it went to court, it was down to 6 counts. He was charged with 6 counts of animal abuse, and that it was thrown out in court and everything was dismissed. But just having that case go to court put PETA on the map, because Alex Pacheco then joined with Ingrid Newkirk, who was the founder of PETA, and together they then became PETA. And Ingrid Newkirk, you may be familiar with, had the famous quote that a rat is a pig, is a dog, is a boy, they're all animals. Meaning basically you shouldn't do anything to a rat that you wouldn't do to your son or daughter or whatever, which again, I don't necessarily agree with. But PETA is big. They do demonstrations against factory farming, so the veal and the chickens that are in the small enclosures, against fur farming. They're famous for going to the fur shows in New York and spraying blood on people, or having their models there and scantily clad, nothing but a sign covering them. And so they're very sensationalistic. They get celebrities like Bob Barker and Pamela Anderson and that sort of thing to spread their message. Interestingly enough, they are the ones who will closely monitor the facility inspection reports. And in fact, this picture, and again, they tend to use pictures from the 1950s and 1960s before there were any regulations. This monkey is from way back in the 60s, and yet this was on a press release that was released just September 1st, 2010. So you can see that they use misleading information in their press releases. They also have a $33 million annual budget, so they have a lot of clout as far as they can get their word out, which is unfortunate because a lot of researchers, unfortunately, don't counteract, but we'll talk about that in a second. There are some active research groups. Interestingly, just as a side note, PETA was taken to court 2 years ago because records show that they had euthanized over 25,000 dogs and cats in the last 8 years and only found homes for 200. So that brings to question their actual mission. One other group that's in the headlines a lot is the ALF, or the Animal Liberation Front. This group started in the early 60s in England. They started out by actually sabotaging deer hunts, first by scaring away the deer and then by slashing the hunters' tires and that sort of thing. They've slowly spread, and now they're active in more than 40 countries. This particular group does not have any ringleader, so to speak, but there are a whole bunch of little disjointed groups that have communications back and forth. Their tactics basically focus on rescuing or liberating animals, and so they're the ones that go to the fur farms in Michigan and Oregon and set all the animals free. Of course, the animals have never lived on their own before, and most of them die or come back to the farm because they want food. And they also use tactics where they use fire bombs, destroy facilities, destroy data, that sort of thing. And in fact, they are on the Homeland Security list as being a terrorist organization. And notably, they do receive money from part of PETA's $30 million. I think it's kind of ironic. They've got their little logo here in the clouds, and it looks like he's got a pet dog and a pet rabbit with his bolt cutters. And so I'm not sure how free his animals are, but essentially that's what they believe in. And so finally, there are some pro-research groups, fortunately, for people on our side. The most well-known is the Foundation for Biomedical Research, was founded in 1981. And basically, their mission is to help promote public understanding and support for humane animal research. They create a lot of educational material that they give to schools and universities and animal facilities that they can post to show that animal research is doing good. There's another group called IFAR, which is Incurably Ill for Animal Research. This is an interesting group. These are actually people that have terminal diseases that have no cure. And so they're essentially fighting for their life. They spend a lot of time on Capitol Hill lobbying to make sure that the funding continues for animal-related research, because essentially, if the funding stops, that's a death sentence for these people. And finally, there's a group that was just formed in 2009 called Protest. This was formed by a UCLA investigator, David Jinch, who's a associate professor in the psychology department studying, I believe it was schizophrenia in primates. And so the animal rights people got a hold of that. They firebombed his car. They threatened his family. And so when Dr. Jinch heard that they were organizing a UCLA protest rally, he got together a bunch of researchers and students that do support animal research and had them march alongside the animal protesters. And they certainly made their point. They actually outnumbered the animal protesters at that rally. And so they're calling for an end to researcher harassment and for an education of the protesters. And there should be more of these people out there, and I applaud him for his efforts. So in summary then, using animals in research is a privilege, it's not a right, and it should be considered that way when you're working with animals. There are federal, state, and local regulations that strictly govern the use of animals in research, and they must be abided by. There is no nationally accepted ethical code regarding the use of animals in research, and it's up to the Institutional Animal Care and Use Committee to set the institutional policy, and they are the ones who would adjudicate any ethical issues that might arise. Every person has their own opinion on the utility of animal research and the applicability of their results, and there is no right or wrong answer. And finally, Currently, at least in my opinion, and for the foreseeable future, the advancement in many areas of science still requires the use of animals. Thank you.